Welcome, everyone. I'm Mo McCarthy, president of the Balmain Institute, and I welcome you to our sixth webinar presentation. This continues our commitment to providing informed discussion of critical issues to an expanded community now brought about by the Zoom technology. We know that you have innumerable opportunities to watch Zoom webinars, and indeed you may be quite zoomed out. And so we deeply appreciate the fact that you've chosen to tune in with us tonight. We are quite privileged and pleased to have as our speaker tonight, Dr. Professor Sarah Palmer. She is an experienced virologist in the area of HIV virus, and she is now centrally placed to consider and concern herself with the COVID virus here in Australia. Sarah will be updating us on the latest research on the coronavirus and the prospects of a vaccine being realized in the near future. A few housekeeping items. The talk tonight will be recorded, and a YouTube video link will be provided to you in the next VI MailChimp. So do look out for it and encourage your friends to watch it as well. The Q&A function on the bottom of your computer will be used tonight for you to type in your questions to ask Sarah, and we will try to handle all of them at the end of the presentation, time permitting. If you have any technical difficulties, please use the raise hand function and someone will come to give you technical support. I now like to turn the meeting over, on behalf of the Balmain Institute, I would like to acknowledge that we meet on the lands of the Wangal people of the Eora Nation, traditional owners of the land in Balmain and Birch Grove. We pay our respects to their elders, past, present, and emerging, and to the traditional owners of the lands of our audience as well. These lands were, are, and always will be traditional lands, never ceded. May we walk these lands gently and in peace, and may we support the recognition of the Uluru Statement of the Heart, and may we be ever mindful and remember that Black Lives Matter. Introducing our speaker tonight is Angela Kim, a member of our BI Executive Committee. Angela, welcome, and please go ahead.
Hello, everybody. Thanks for tuning in. I'm Angela Kim. Tonight, we're privileged to have Professor Sarah Palmer speak. This year has seen the worst pandemic since HIV came to light in the 1980s. It's caused devastation to our liberty, our health, community, and the economy. Professor Palmer is the co-director at the Centre for Virus Research at the Westmead Institute and professor at Sydney University Medical Faculty. She is a world-renowned researcher in HIV, and her work identifying HIV proteins that elicit an immune response is being used in the search for a COVID-19 vaccine. After the talk, there'll be a Q&A, and you'll be able to type questions into the Zoom app, and I'll read the questions out to Sarah. Now I'll hand you over to Professor Palmer, who will speak to us about COVID-19, its mutations, its long-term health effects, and the quest for a vaccine. So over to you, Sarah.
Thank you, Angie. And welcome everyone for joining our discussion about the COVID-19 pandemic. And hopefully some of our discussion will convince you that possibly we can contain this pandemic. But first, I would like to start by paying my respects to the traditional owners of the land and their elders, past, present, and emerging. Absolutely, Black Lives Matter. So some of the most important global statistics are shown here in this graph. Now, I don't know how you are at home, whether your families have seen a decrease in shaving or a decrease in bra wearing. And for that matter, you don't even know if I'm wearing sweatpants because Zoom allows us to do this. But one of the statistics that are shown in this graph is quite important, and that is the confusion over mask wearing. At the beginning of this pandemic, people were very confused as to whether they should wear a mask. And then it even came, they didn't wear a mask at the beginning of this pandemic. And then they became quite confused as to whether they should wear a mask. And then as the pandemic went on, they started to wear a mask. And wearing a mask is very, very important because this virus is extremely infectious. In fact, one person has the ability to spread the infection to 5 people. And these 5 people have the ability to spread the infection to 125 people. So there is an exponential growth in infection from one infected person. And this is what has led to the pandemic of the SARS-CoV-2 virus, which causes COVID-19. So why is this virus so extremely infectious? Well, we first have to turn to the little virion, which is shown here. And this is a viral particle of SARS-CoV-2, which causes COVID-19. And most coronaviruses look like this, where we do have 4 seasonal coronaviruses, which cause the common cold. And they all have this spike protein on their surface. And this spike protein allows this virion to actually attach to the cells that it targets and infects. And this spike protein allows this little virion to attach to those cells and infect them. But why is SARS-CoV-2, which causes COVID-19, why is this virus so much more infectious than the seasonal coronaviruses? And that comes down to these protein assessment and the genetic changes that have been found in this virus. And that is, when we looked at the spike protein, it was found that the spike protein of SARS-CoV-2 has this little flap. And this little flap allows this spike protein, which actually binds to the receptor of the cells that it targets and infects, very tightly. So it actually brings the virion and the virus closer to the cells and allows the virus to infect the cells much easier. So that is why this virus is much more infectious than the seasonal coronaviruses. So, and that is why wearing a mask is really important as well, because this is a very infectious agent. So how is this virus transmitted? And there's many different ways that the virus can be transmitted, but predominantly it is transmitted by the spread of small droplets, small respiratory droplets that are caused from sneezing, coughing, and talking. And so these small droplets can be spread in the air, and the virus actually takes advantage of that, and that's how it infects goes from one person to another. And if you look at this figure here, standing 1.5 meters from each other prevents a lot of these droplets from allowing you to be exposed to a lot of these droplets, because the majority of these droplets will drop to the ground prior to 1.5 meters. That's why maintaining a distance of 1.5 meters is so important and prevents new infections. Furthermore, wearing a mask actually prevents the droplets from traveling even further and keeps the droplets very close to a person who could possibly be infectious. Secondly, people can be infected by touching infected surfaces and contaminated surfaces, and then also touching their face. But the virus concentrations on surfaces is quite low, because once the droplets dry, only traces persist on these surfaces. So the actual infection from a surface is quite low, or the percentage of people who can be infected from a surface is quite low. But where the infection is really most important and where we see an increase in infection is indoors, because respiratory droplets can remain indoors for a long time in poorly ventilated environments. And in fact, the chance of becoming infected by this virus indoors increases by 30%. So this transmission can occur through aerosols, and these aerosols that remain in these poorly ventilated environments indoors does increase infection by 30%. Furthermore, wearing a mask is important because there are many people who have an asymptomatic infection. Over 30% of people who are infected by this virus have no symptoms and remain undetectable, yet they do shed the virus and they can infect others. Furthermore, 40% of people who are infected and who go on to have symptoms can shed the virus 1 to 2 days before they have these symptoms. So up to 70% of infections can be, well, 30% can be asymptomatic and 40% can be pre-symptomatic. So about up to 40% of infections can happen even when people don't even know they're infected. So therefore, surgical masks are very, very important to wear, especially where there's a high incidence of infection. But one thing I would like to stress about surgical masks is that they block the source of infection better than the reception. So they will prevent these droplets from being aerosolized, but they are not as good as blocking the reception of the virus. Furthermore, when people take off their masks, they can, if they don't do this in a careful way, they can also infect themselves. So what are some of the symptoms and signs of COVID-19? Well, predominantly this virus, when it attaches to the cells that it targets, these cells line the respiratory system, both the upper respiratory system and the lower respiratory system. And because this virus can infect these cells quite readily, it causes a very serious respiratory infection in some people and a mild infection in others. And in a mild infection, we see predominantly sneezing and coughing, runny noses and shortness of breath. But in more severe cases, this infection can cause pneumonia. So predominantly it's a respiratory infection, but in more severe cases, and especially in those who are over 60 years of age, it can cause a more severe infection. Why is that? Well, that is because our immune system, as we age, functions, it starts to function less well or poorly, becomes less functional. And in some of people who are elderly, when they become exposed to the virus, they have a delayed immune response. And this delayed immune response allows this virus to cause pneumonia. But after some period, the body senses that there's an infection. And this can lead to what we call a cytokine storm, where the body sends out signals to the immune system saying, I'm infected. And a lot of cells then rush to the lung. And this is what we call a solid lung, when a lot of white cells rush to the lungs to fight the infection, and it can cause a solid lung. And this is what can cause severe acute respiratory syndrome, because lungs become inflamed and congested. And that's when a lot of times people who have this severe infection have to go on ventilators. And this is a microscopy, electron microscopy showing the virions infecting lung tissue. So the red dots here are the coronavirus, and they're infecting the lung tissue. And as you can see, they can spread quite rapidly or freely throughout the tissue. But there are other side effects of this virus that are still being understood. And that is that this virus can cause other symptoms and signs, such as severe kidney failure. They can cause clotting, and we believe that's due to a lot of inflammation in response to the virus, can cause high levels of clotting in some people, which lead to cardiovascular damage and also strokes. So this virus has a lot of different signs and symptoms, which we're still learning about, and can cause very severe disease and also can lead to death in about 3% of people who become infected. So as I described, about 3% of people can die from this infection. But if we look at the infections, the pyramid of infections overall, 81% of people experience a mild case of infection. And these are patients who may not have pneumonia or have mild pneumonia. And then we find that 14% of patients can have a severe case. They have decreased white blood cells, shortness of breath, and they can have these lung infiltrates, which are the white blood cells coming in to fight pneumonia and fight infection. And then 5% of infections can lead to a critical case. And these are people who can suffer respiratory failure and also organ failure. And about 3% of these critical and severe cases can lead to death. And there's some pre-existing conditions that can lead to a higher rate of death. And these pre-existing conditions include cardiovascular disease, diabetes, chronic respiratory disease, hypertension, and cancer or being immune compromised, such as HIV infected individuals. A lot of these pre-existing conditions do happen as we age in life. So that's what also leads to more severe and critical cases in people as they age. And as I have discussed, that this is a very serious disease for those who are aging. And as we can see by this graph, that mortality greatly increases as a person ages, to about 15% of people who are 80 years or older can die from infection from COVID-19. This data came from China, but it's also been shown in the United States, very similar statistics, that as we age, the mortality rate increases from COVID-19 infection. So how can we control COVID-19? Well, we are still waiting for some very effective antiviral drugs. However, there is one antiviral drug that is available, and that's called remdesivir. It was developed by Gilead Sciences, and it is shown that this virus can shorten the duration of hospitalization by 31%. But currently, that is the only antiviral that we have available for fighting COVID-19 infections. I do want to point out that hydroxychloroquine, which is used for malaria, is not effective against COVID-19. And both the US NIH and WHO have stopped their trials of this compound. Some anti-HIV compounds were used to see how well they would reduce infection, and they failed in clinical trials. We do have currently some of the treatments that are being used include convalescent plasma therapy. And this is when plasma from patients who have recovered from COVID-19 and thus carry protective antibodies are injected into patients who are newly infected with the virus. Unfortunately, just recently, in the last day or two, a study has come out from India showing that convalescent plasma therapy is not as effective at reducing viral infections and actually reducing the severity of COVID-19 infection. And in fact, they found that even in convalescent plasma, there's very low levels of antibodies. So they're not that protective for patients in preventing infection or reducing the severity of infection. Studies have shown that steroids can help reduce the inflammation from the immune response to this infection and can actually reduce the risk of dying within 28 days compared to standard care or placebo. So using steroid treatment can reduce some of the side effects of inflammation. So how else can we control COVID-19? Well, right now, what we have in our toolbox is quarantine and social distancing. This does sound old-fashioned, but social distancing and wearing of masks and washing our hands does help to prevent infection from COVID-19. And especially for healthcare workers, having adequate personal protection equipment is very, very key to protect our healthcare workers who are at the front line from being infected from COVID-19. However, over 100 groups now are developing vaccines. And these are currently in development. 3 of these vaccines are in development in Australia. But most of these vaccines are aimed at younger populations. They'll be most effective against the younger populations. And as we've seen, it is the elderly that have a higher risk of mortality from this vaccine, from this COVID-19 infection. So we do need vaccines that are effective for those people who are older. And there could be bottlenecks in the scale-up of these vaccines, but we do know that over 100 groups are developing vaccines, and hopefully some of these vaccines will be effective for patients to prevent infection in people who are over 65 years or even older. So we've heard a lot about herd immunity, and herd immunity has been proposed as one way to protect people. But I would like to talk about herd immunity in the regard in the impact of a vaccine and how that will induce herd immunity. So when we look at herd immunity, there's three models of herd immunity. And the first model, and that's where we are today, is where the majority of people are not immunized, do not have any immunity or natural immunity to this infection. And that's where we are today with COVID-19 because it is a new virus. And so when one person is infected, they can infect a lot of different people. And essentially, society becomes infected because there's no immunity present. And we all are new to this virus. The second model of herd immunity is when we have a vaccine that's either 50% effective, or we have a vaccine that comes available and it's given to those people who are on the front line, such as healthcare workers and those who are working on the front line. And that's when some people have been immunized, but the majority of people are still not immunized, and they are actually susceptible to infection from the virus. So again, we see a large spread of infection throughout society. And the third model of herd immunity is where we want to be today. And that's where the majority of people either have natural immunity or have immunity that's been enhanced and instigated by vaccine and immunization. And that would be where the majority of people have now natural immunity due to vaccination. And then the majority of the society is then protected from infection. And by the majority of society having immunity, they are not susceptible to infection from COVID-19 or from a virus. So currently we're in the first model, but where we would like to end up is the third model of herd immunity. So we have heard that there are some countries, predominantly Sweden, who have thought that possibly allowing their society to become infected and develop natural immunity would be one of the best ways that they could actually protect their society. In other words, enhance a natural herd immunity. But in order to have a natural herd immunity, 60% of the population has to have antibodies. And so 60% of the population has to be exposed to the virus and have produced antibodies. But with this virus, if 60% of the population has been exposed, that means there has been a lot of death also, because this virus does have a 3% death rate. And this natural herd immunity is not working really, because when we look at New York City, where there is high infection rates, only about 20% of people developed antibodies. In Stockholm, it's 7%. In London, it's 17%. So none of these cities have reached the level of herd immunity, which would actually protect a society. So herd immunity may not be the best solution for COVID-19. Furthermore, to develop 60% of herd immunity, we would have to experience a lot of mortality and death. So we realize that when patients get infected by this virus, or people get infected or exposed to this virus, it can be up to 2 weeks before they really show that they are infected, and that it can be up to 2 weeks before tests can actually determine that people are infected from the virus. And also we have a small percentage of people who never really show any symptoms, so they wouldn't even know that they were infected. But it takes about 2 weeks in order for the test to be absolutely positive once a person can be is exposed to the virus. So for some people it's shorter, it can be 2 to 3 days, but for others it can be 12 to 14 days before they really show signs of the virus and then go to be tested and are found to be positive. But what we don't know is about antibodies. We know that when patients become infected, they do develop antibodies. But what we don't know about these antibodies to COVID-19 is how long do these antibodies persist? And studies have recently shown that in 40% of asymptomatic patients and in 13% of those who experience symptoms, that within 8 weeks, antibodies drop to undetectable levels. So we're not really sure how long antibodies will persist in patients or in people once they've been infected with COVID-19. And furthermore, this is a bit worrisome for vaccines, but that does lead us to wonder how long or how many boosters we'll need from a vaccine, or how long a vaccine will actually elicit an immune response, and how many vaccinations a person would need to really elicit a long-term response to this virus. So in talking about the vaccines, there are over 100 that are being developed, and all the vaccines that are in development are actually being developed against the spike protein. So all the vaccines contain an element of the spike protein, whether it's RNA or DNA or a combination or the actual protein itself. All of the vaccines are being developed today contain some part of this spike protein. So that is the major part of the virus that they will be targeting or actually eliciting immune response to, is this spike protein. But the question we get a lot of times in the field is, why does it take so long for vaccines to be developed? Well, there's a lot of different steps to developing a vaccine, and clinical trials are needed. And furthermore, once a vaccine is developed, it takes a long time to ramp up the manufacture of this vaccine, the administration of the vaccine, and then also for people who have been vaccinated to develop immunity. So let's walk through sort of the phases of clinical trials that are needed for a vaccine to be developed. So the vaccine testing process goes like this. First, vaccines have to have preclinical testing, and that's where new vaccines are tested on cells and injected to animals to see if an immune response is elicited either in cell culture or in animals. And once the vaccine shows that it does elicit an immune response, it can move on to phase 1 safety trials. And this is where a vaccine is given to a small number of people to test the safety and dosage of the vaccine. And also the tests are run to determine, does this vaccine stimulate the immune system and elicit an immune response towards the virus? Once a vaccine passes the safety trials, it can move on to expanded safety trials. And this is where the vaccine is given to hundreds of people, typically split into different groups of children and elderly, to test whether the vaccine is effective, safe, and whether it elicits an immune response in these two groups, in children and in the elderly. If a vaccine is found to be safe and elicits an immune response in a majority of people in a phase 2 trial, it will move on to what we call the efficacy trials or phase 3 trials. And this is where a vaccine is given to thousands of people, up to 30,000 to 40,000 of persons, to determine how well it can elicit a response in these people, but also to determine if there are any rare side effects from this vaccine. And this will be very key for the COVID-19 for any vaccine against COVID-19. Because we will be giving this vaccine to tens of millions of people. And if we have 1% of these people to have a serious side effect, that will be a lot of people. So it really is a key that COVID-19 vaccines go through very serious phase 3 efficacy trials, and that they are tested in thousands of people. Finally, if the vaccine passes this efficacy in phase 3 trial, it can be approved by regulators, and some vaccines can receive emergency authorization to be used immediately, especially for healthcare workers and those at the front line. The vaccine can be given to people to those who are most at risk, and be recognized as an emergency use authorization. Now, some of these phases can be combined. Sometimes when testing a vaccine, you can combine phase 1 and 2. In other words, start to give the vaccine to a small group of people and very quickly ramp this up to hundreds of people. And also phase 2 and 3 phases can be combined so that vaccines can be approved quicker. But still, it is a very long process, and it's very important to go through this process in order to determine whether a vaccine elicits an immune response and also is safe. When we talk about vaccines for COVID-19, there are 4 types of vaccines. The first type is where actually RNA and DNA that represent the spike protein. So the RNA and DNA that actually can develop into the spike protein are vaccinated directly into a person. And these genes will then elicit an immune response towards the spike protein. Right now, there are several vaccines that are in phase 1, 2, and 3 trials, and there's only one that's in phase 3 of a RNA vaccine, which is being developed at the US NIH in collaboration with a company called Moderna. And right now they are enrolling 30,000 volunteers across 89 sites in the US. And so they are in phase 3. And this is an RNA vaccine, which once injected directly into a volunteer or a person, will elicit immune response towards the spike protein. There are viral vector vaccines, and these viral vector vaccines these viral vectors contain both RNA and DNA of COVID-19, and the viral vectors are adenoviruses, which actually can bring the RNA and DNA directly to cells. And again, the cells will start to make the proteins from the spike protein from the RNA or DNA, and this will elicit an immune response. What I should point out that because this is a small fragment of the virus will not replicate, because both these RNA and DNA vaccines and the viral vector vaccines that contain the RNA and DNA of the spike protein, they're just a small portion and small protein of the virus, so the virus will not replicate. But the immune system will recognize that these are foreign elements and will elicit an immune response. So to viral vector vaccines, we have several that are in phase 1, phase 2, and 3 that are in phase 3 trials. The one I want to bring your attention to is the Oxford University and AstraZeneca vaccine. This is an RNA adenovirus vaccine. It was being tested in England, Brazil, South Africa, and the US, but there was one severe side effect from this vaccine during this phase 3 testing. So right now, the testing has resumed in England, but has not resumed in Brazil, South Africa, and the US. The side effect was inflammation of the spinal column of one of the volunteers who received this vaccine. So they have resumed the testing of the vaccine in England, but the US and other countries have not resumed the phase 3 testing of this vaccine. There are also protein vaccines, and these are vaccines that use just the coronavirus protein. Again, this is the spike protein, and the fragment of this protein is usually used to provoke an immune response. And there are several clinical trials that are in several vaccines that utilize the protein of COVID-19 that are in phase 1 and phase 2. And then there are several vaccines that use sort of the old traditional method where we use the whole virus vaccines. And these are vaccines that use weakened or inactivated viruses or inactivated COVID-19 to provoke an immune response. And there's several vaccines that are in phase 3 that are using inactivated and weakened COVID-19 as their vaccine. I did want to point out one other vaccine. I'm sorry, that's part of the viral vector vaccine, and that is the Sputnik vaccine that's being given to 40 volunteers in Russia. Studies of this vaccine are a bit concerning because the data that just was published looks quite suspect. So we don't really know how well this vaccine is working, to be honest. And that's quite concerning because it's being given to 40,000 volunteers in Russia. And in fact, Putin said that he injected his daughter with this vaccine. So stay tuned. So we do have several vaccines that are in phase 3, but to be honest, we still have a long way to go, because even if they pass phase 3 testing, they still have to be manufactured and then disseminated. But there are still some challenges to the vaccines. And the major challenge to the vaccines that are being developed today is that this virus is changing genetically. And what do I mean by that? Well, what I'm showing here is what we call a phylogenetic tree. And each one of these small green dots is a sequence, is a full genome sequence of the COVID-19, SARS-CoV-2 virus, or COVID-19. And so each one of these small green dots is the viral sequence. And what we can see is that in this part of the tree, and the branches, so this green dot is the genetic diversity between this green dot of SARS-CoV-2 to another virus is the length of these branches. So the genetic changes from this virus to that virus is based on the length of the branches shown in this phylogenetic tree. So what we can see is that there's a lot of green dots, and these green dots represent the original virus that was caused the infection in Wuhan, China. And if we look down at this map, we can see there's a lot of green shown, that this is where the original, this green virus, the green dots represent the virus from Wuhan, China, and are shown in the map here. But one genetic change from the Wuhan virus has caused the virus to become what we call more infectious. And that's represented by these yellow dots. Just one genetic change from the Wuhan virus in the spike protein has caused the virus to be a lot more infectious. And as we can see, there's a lot of these yellow, there are a lot of these yellow dots, which represent the virus as it spreads across the world. And this more infectious strain of the virus is spreading across the world. So if we look at the United States, 3/4 of the infections in the United States contained this one mutation in the virus that has made it more infectious. And the worry that I have about this change in the virus is that the majority of the vaccines have been developed against the Wuhan strain. And whether these vaccines will be effective against the strains that are spreading now throughout the world remains to be seen. Again, as I showed before, the virus is changing rapidly. So the Wuhan strain is shown here, but each one of these dots and the colors of these dots are shown where they were originated. And as you can see, there's a lot of different genetic change across this virus. And this genetic change within this virus, as it starts to spread across the world, will be difficult for vaccine development. And furthermore, there was a reinfection case in Hong Kong. And this was a person who was infected in Hong Kong in March, and 4 months later, it reinfected in Europe. And this also indicates that the first infection did not produce enough immunity to prevent this person from being reinfected by a second strain. And this also brings some concern for vaccines, because if the vaccines are developed against the Wuhan virus, will they be effective against the strains of the virus that are now forming as the virus goes through the world? And this is a sequence analysis that we did that a very talented researcher, John Sebastian Eden, in our group, in our center, conducted, and he sequenced the virus in New South Wales. And this is, again, another phylogenetic tree. The Wuhan strain is found here. It's that black dot. That's what the original prototype of the virus found in Wuhan. And as you can see, the virus is continuing to change. And the strains that are found in New South Wales, which are represented by these red circles, are very genetically diverse. Again, indicating that a vaccine towards people who are infected in New South Wales will have to be effective against multiple different strains of this virus. Furthermore, we can see that some of these viral strains group very closely together. So this is the Wuhan virus, but these are the strains found in the United States. And they group closely together, but they still are genetically diverse and have genetic changes across them. So this does bring us to several questions about how effective our vaccines will be going forward. So serious questions about the vaccines are: 1, how effective will vaccines be for all ages? Will the vaccines developed from the Wuhan strain be protective against newer strains of COVID-19? Will it require more than one injection to develop immunity? And will it require a yearly injection similar to the flu vaccines? So these are all serious questions we have to address when thinking about vaccine development towards COVID-19. And one last slide topic I would like to discuss with you is how the SARS-CoV-2 pandemic is affecting other pandemics, such as the HIV pandemic. I originally have been doing most of my research on HIV, and I am now doing some research on COVID-19. But the effects of this new pandemic on the existing HIV pandemic can be quite severe. We are seeing that there's interruption of anti-HIV drug supplies, poor clinical care, and suspension of HIV testing. And all of this will lead to higher mortality rates, particularly in developing countries for HIV, for the HIV pandemic. Furthermore, we've seen interruption in condom supplies and peer education. And this could actually lead to the increase of HIV incidence in developing countries and in all countries, to be honest. So there will be effects of the SARS-CoV-2 pandemic on the existing HIV pandemic. So that is my presentation for tonight, and I look forward to your questions, and possibly there are some topics I haven't covered, so I look forward to your questions. Thank you very much for your attention.
Thank you very much, Sarah. Thank you so much. I've got a question here from Richard Bouvrier. He says, if say 1 million have died of COVID in 5 months, 200,000 per month, why is it so important to go through the slow, old, uni-style drug process?
The drug process? You mean developing antiviral drugs?
I think he might mean the vaccination. Going through the
very
vaccination
important to go through clinical trials and to test both drugs and vaccines, because we will be using these compounds against tens of millions of people, within tens of millions of people, and it could cause very serious, if there's any serious side effects, that could cause even more damage to people. So we really have to conduct very serious clinical trials to ensure that that we're not harming people more than helping them against this pandemic and this virus.
Thank you. Is there research on the incidence of long-term effects on the brain and heart in people who recover from the respiratory illness?
So there is research ongoing, and there have been studies that have shown that there is scarring to the lungs of people, even people who have had a mild or even a mild infection. They've found that there is scarring of the lung tissue, even in athletes, high-performing athletes who've had a mild infection. They have found there is scarring of lung tissue. And also there are very serious difficulties in neurological confusion happening long-term in some patients. So there are some long-term effects of this virus. And these studies are ongoing as we speak to determine what are the long-term effects, because this is such a new virus. We're new to this virus and we're just learning about it.
And another one, what do you think about Sweden's response to COVID-19?
I'm not a big fan of Sweden's response to COVID-19. First of all, you can just look at the death rate in Sweden. They have 5,000 deaths because they did not go into a very severe lockdown. if you compare that to Denmark, a country that has similar population, they have about 60 deaths. So Sweden has paid a very high price for not instigating a more serious lockdown. Now, the Swedes themselves, some of them are very supportive of how their government has handled the pandemic, but it will remain to be seen what happens now as we go into winter, where people are more are inside, and whether the spread of the virus will, there'll be a spike in the spread of virus in Sweden and leading to more mortality.
And do the phase 3 trials have to be conducted in populations where there is a high incidence of COVID-19?
Yes, that's absolutely true. Phase 3 trials must be conducted in countries where there's a high incidence. So that way we can determine whether they really are effective at preventing new infections. So for example, Australia may not be the best country to conduct a phase 3 trial. That's why when you look at those countries that are conducting phase 3 trials, it's predominantly United States, Europe, Russia, Asia. So yes, there has to be a high incidence level to really test the efficacy of these vaccines in a phase 3 trial.
And from Margaret Wallace, if a friend and I both get vaccinated, can we hug each other, or must we always be 1.5 meters apart?
If the vaccine is effective in eliciting natural immunity, go ahead and hug your partner, hug your friend, as long as they're vaccinated. But really, it really is important that the vaccine elicits natural immunity and long-term natural immunity. And that's something we'll have to learn about, whether we'll need boosters. They're already talking about the AstraZeneca vaccine. We may need a booster. The Moderna NIH vaccine, we may need a booster. So really, it will depend on how long immunity lasts once we receive a vaccine and whether we need a booster to continue to protect ourselves and to protect the community around us.
What makes some people more susceptible to severe effects of COVID-19? Would it be best to find a vaccine that protects the people most at risk of high morbidity?
Yes. It would be. And that's something we're working on right now at the Westmead Institute. We're working on a second generation vaccine in which we hope will be more effective against the people who are older and those who have comorbidities or failing immune systems such as HIV infected individuals. So yes, it will be very important. But right now, the vaccines that are being developed are being developed for those who are younger predominantly. And I think that's because there's a high incidence in those who are young as well. But yes, we do need vaccines to be developed for those who experience a more severe disease. However, if we can vaccinate people, especially young people, and a majority of them elicit an immune response, that will be somewhat protective of people who are older, but still we need vaccines that are targeted for people who are older. And our vaccine, we're including an immunostimulant, which would have to help to stimulate the immune system, because as we age, our immune system doesn't function as well. And so in the vaccine that we're developing at the Westmead Institute, we will include an immunostimulant.
And another question from an anonymous person. Surely if 200,000 are dying per month, in order to morally justify adverse incidence argument you utilise, wouldn't they also have to number around a mortality of 200,000 per month? And he's quoted Aristotle, compare like with like.
So I guess they're talking about Sweden, I'm not sure. Those 200,000, those 200,000 deaths per month are worldwide. So, and in some populations we have a more elderly population. Also, as elder care centers where the virus was introduced, there was a higher mortality rate than 3% in those centers. So it really depends where the virus was introduced as to the mortality rate.
And from Gail, what do you think is a realistic time frame for a vaccine to be available here in New South Wales? And what about the mutations by the time the vaccine is developed?
That's an excellent question, Gail. We are very worried about the mutations because this virus is still going through multiple populations, multiple people, and it's changing. So we will most likely have a vaccine, and I'm hopeful for a vaccine in 2021 in the second and third quarters of that, or third and fourth quarters, to be honest, of next year. But we will have to keep watching the virus and and sequencing the virus so that we understand what strains we really have to target and what changes we have to make in the vaccine so they will be effective against the virus that we have today, as opposed to the virus we had 6 months ago.
And from Warwick Reed, could we ever get COVID under control globally without a vaccine?
Well, you know, just yesterday, the director of the CDC from the United States said that mask wearing may just be as effective as a vaccine. And I think, what he was referring to is that if immunity is not elicited in a large proportion of people, then we will have to continue to wear masks to protect society. So if everyone wore a mask, if we practiced social distancing, if we washed our hands continually and used sterilizing reagents for our hands, yes, we could most likely knock the incidence down to a very low level. I mean, we can point to countries such as South Korea, New Zealand, even Australia. Australia has done very well. If you look at New South Wales, that by practicing and maintaining these practices, that's the difficulty. Maintaining these practices for the long term, it can be difficult, but I do believe that by maintaining these practices, we can decrease the incidence of COVID-19.
And from Zoe, I'm a healthcare worker and have come across young people with significant COVID-19 morbidity, prolonged recovery, post-viral fatigue. Is it known what makes some of these young people so much sicker?
Yeah, it's not known yet, but there are some genetic factors that have come to the forefront that some people have a genetic predisposition, I can never say that word, a genetic a genetic predisposition to become what we call a long hauler. So a long hauler are people that were infected with COVID-19 but are experiencing symptoms for a long term. And we do believe there are some genetic relationships to that. Yeah, and then there are the pre-existing conditions that can lead to more severe and long-term effects of this virus.
And from Gail, will it eventually be like the seasonal flu vaccine then, changing each year to deal with the mutations?
Gail is absolutely right. That's how I see it. I think what we will eventually have is a flu vaccine and a COVID vaccine together, and we receive it every year because this virus can continue to mutate. It's an RNA virus. It's very easy for this virus to mutate. So I see it that way. I totally agree with Gail that we will most likely have to have a yearly injection for COVID-19 or for the next iteration of COVID-19. And I also believe that the first vaccines, we may need multiple boosters in order to enhance immunity towards COVID-19 that we have today.
And we've run out of questions now. So thank you very much.
Thank you.
That's a wonderful talk. Thank you.
Thank you very much for everyone for listening.
And I'll hand you over to Mo.
Thank you.
Thank you so much, Sarah, for that interesting and stimulating presentation. It's very clear that COVID is still a threat to us, and we all need to take care in what we do, particularly wearing masks, observing social distancing, and washing our hands. Before we close, I would like to thank everyone who has donated to the BI for these webinars. They do incur their own costs, and we deeply appreciate what people can give, realizing for a lot of people it's a very difficult time. So we appreciate anything you can give as you can. And if you can't, well, that's all right too. We hope you've just enjoyed the webinar. The bank details for the BI are located at the bottom of your MailChimp announcements of the speakers. And you can then just transfer funds directly to the BI account. As always, I'd like to thank Michelle French for her wonderful technical support in making these performances so problem-free, and also to Stephen Burstow for taking care of and troubleshooting any of the technical problems that have come up during our presentations. Our next speaker will be Bruce Volpe, who is speaking on an update on the upcoming US elections. Even if you're not an ex-American like I am, you probably have some interest, if not a great deal of interest, in what happens to this election on November 3rd in the US. He will be speaking on that topic on the 8th of October. And please, we hope that you'll plan on joining us then. The announcements will be coming out closer to the date. So in closing then, stay safe, wash your hands, wear your mask, keep your social distance, and take good care. Thank you for tuning in. Thank you again, Sarah and Angie. Have a good evening, everyone. Good night.